Thank you very much. So I want to take you today for the next half hour on a global tour with me of some of the research we're doing on the cognitive neuroscience of learning and generalization. And this half-hour tour begins 2,000 years ago in ancient Greece, where Aristotle asked the question, how do we come to associate two things in our mind? And Aristotle came up with 3 rules of association, which are critical even to this day for understanding research in both psychology and neuroscience of learning. His rules were 1, contiguity, experiences near each other in time or space come together. Frequency, experiences that co-occur often are associated together. And lastly, similarity, experiences similar to one another are associated. Now, Aristotle's rules of association were extended to become theories of learning. Let me just start with some definitions. Learning we define as the process by which changes in behavior arise as a result of experiences with the world. So an example is Aristotle tries broccoli for the first time. It makes him sick. He develops an association, broccoli sickness. So he's learned that, perhaps from 1 or 2 trials. But the real challenge then is generalization, the transferring of past learning experiences to new situations. Aristotle has to know whether or not, when he comes to cauliflower for the first time, does he eat it or avoid it? Does he discriminate or generalize? So the work in our lab builds on learning and generalization to ask 3 questions that span 3 fields. How do people learn to associate 2 things in their mind? What brain systems contribute to learning and generalization? And how does understanding the brain systems for learning and generalization inform the diagnosis, treatment, and prevention of brain disorders? And we work with these 3 questions at 3 different levels, levels of the brain, of behavior, and clinical applications. And the glue which ties together these levels are computational models, which allow us to understand how principles at one area relate to principles in the other areas. So the work I'm going to talk about today, I'll start by describing some of our ongoing research in 2 main threads, one looking at associative learning in the striatum, the other at generalization in the hippocampal region. And then in the second half of the talk, describe 3 new integrative research directions that combine those work in some new areas. So let me start by talking about the associative learning, the striatum, and Parkinson's disease. So let's go back to Aristotle, as we will throughout the talk, and note that 2 of his principles, contiguity and frequency, are the basis for modern theories of learning. In particular, theories that trace themselves back to Thorndike, who studied how cats learn to escape from boxes, and he developed the law of effect, he described. And the law of effect has 2 parts. He says behaviors with positive outcomes, that is reward, are increased, and behaviors with negative outcomes or punishment are decreased. And if this theory of learning sounds familiar, it's because he borrowed it from another scientist, from Darwin, and basically took Darwin's theory of natural selection across generations and applied it to principles of learning within an individual. Now, this law of effect distinguishes between learning by the carrot or the stick, as we say colloquially. There are 2 ways to motivate the mule. It can learn to approach a reward, or it can learn to avoid a punishment. And those of you who've raised children know that there are a lot of individual differences. Some children respond better with a treat if they do the right thing, others with a threatening of a timeout or other punishments if they do the wrong thing. And it's those individual differences in whether people are prone to approach reward or avoid punishment that are the basis for some of the studies that I'm going to describe now. So the behavioral paradigm, it's a category learning task in which learning is both by positive reward and negative or punishment. And in this task, people see various little artistic diagrams, and they have to predict whether they go to category A or B. They'll make a prediction, and 3 things can happen. They can sometimes get positive feedback or reward if they get the right choice. They'll sometimes get negative feedback or a punishment if they make the wrong choice. And in this particular experiment, there are also no feedback trials where they won't be informed whether they made the right choice or not. Now, the structure of the task is as follows. Half of the stimuli in this task are trained by reward, so that if they get a correct response, if they give the correct response, they get a positive feedback. But if they give the wrong response, they get no feedback. And the other half of the trials are trained by punishment only. So if they get the correct response, there's no feedback. And if they get the wrong response, they get negative feedback. So the no feedback trials are ambiguous. And in this way, we can begin to measure whether people are more sensitive to being trained by the carrot or by the stick. And it's these individual differences that we'll measure in a variety of different ways. So where in the brain does this kind of feedback learning arise? A wealth of evidence suggests that the striatum within the basal ganglia, particularly the dorsal striatum, is key for stimulus-response feedback learning. And that gives us an opening to look at this in humans, because this is the brain region that's impaired in Parkinson's disease by the loss of dopamine. So the question that we ask in the research is, how is feedback learning affected in Parkinson's disease? And the challenge here is that most Parkinson's patients that we find when we go to the clinics here around New York and New Jersey are already on some kind of medication, dopaminergic medication. And the challenge is, how do we differentiate the effects of the medication from the effects of the disease? So that gives us the global question, where in the world do we find Parkinson's patients who have not yet been medicated? And this brings us to Hungary, where thanks to sort of the Soviet-style medical system, all patients go through a central medical system. In this case, in Budapest, Hungary, with Szabolcs Kary. We work through the Semmelweis Hospital through their Parkinson's clinic. And the advantage of going to Budapest is I have the hospitality of our colleague Laszlo Zaborski, who lends me his beautiful apartment. You should all do the same. So, in this task, you can see with healthy controls, we look at how they learn from reward or punishment across blocks, usually about 40 trials per block, showing the optimal response, essentially the correct response. And healthy normals, people learn from both reward and punishment. However, de novo Parkinson's patients who've never been exposed to medication show a very selective deficit in learning from reward, but not from punishment, consistent with the idea that dopamine carries a reward-based signal. But these patients are then put on dopamine agonist medication, and what happens is it flips. The dopamine agonists remediate the deficit to reward learning. They're now normal at reward learning, but it induces a completely new deficit in learning from negative feedback or punishment, something which may be related to the tendency for some Parkinson's patients to develop impulse control disorders, where they're insensitive to the negative consequences of their actions. So a question that's left aside is, these are all patients who have Parkinson's disease. Do these cognitive changes precede the onset of motor symptoms? And the problem there is, how do you find prodromal Parkinson's patients, patients who we know are going to get Parkinson's disease, but not for a few years? And again, we go to Hungary. We take a road trip for about an hour and a half to the rural area of Buzsák. And as you can see on the map and from the pictures besides, when it comes time to find a wife, there's not a lot of options there. There are only a few people in the village, so you marry your first cousin. And they've done this for many generations. And as a result, there's a rare duplication of the alpha-synuclein gene that's in a particular family in this village. And we studied these people. And what we found is these people with this gene, which almost certainly will cause Parkinson's, have the same pattern as de novo patients, namely they have a reward learning deficit. And 2 years later, all the people we tested had Parkinson's disease. So it suggests that this reward-based feedback learning precedes the motor symptoms in Parkinson's disease. So let me summarize this first thread. De novo patients, PD patients, and prodromal patients have a selective reward learning deficit, but not with punishment learning. Recently medicated patients on dopamine agonists have a remediation of the reward learning deficit, but an induced deficit in negative feedback. And the striatum and dopamine are key for learning feedback, particularly positive or rewarding feedback. Some of the current directions that I don't have time to talk about are, one, we're looking at functional brain imaging correlates in a project at North Shore, Long Island, where we're part of a P50 Udall Center grant. We're working on looking at the brain imaging correlates of these changes in cognition with medication. In other studies in New York and at Robert Wood Johnson, looking at the links between the cognitive and psychiatric aspects of Parkinson's disease, particularly depression and impulse control disorders, which are common in Parkinson's. So let me now turn and shift to the second thread, looking at generalization and the role of the hippocampal region. So back to Aristotle again, and there was the third notion that he had of similarity, and understanding similarity is the basis for modern theories of generalization. Generalization, to remind you, is the transferring of past learning to new situations. It's how Aristotle decides whether to eat or to avoid the cauliflower based on his experience with broccoli. So Aristotle has the challenge to discriminate or to generalize. He has the cauliflower, and he has to decide, are cauliflower and broccoli sufficiently similar? Are they equivalent enough that I should expect them both to make me sick? Or do I discriminate and decide that they're sufficiently different that I might actually try the cauliflower and like it? So where in the brain is this kind of generalization behavior determined? And one brain region that's critical is the hippocampus in the medial temporal lobe. And soon after I arrived here in the early 90s, Catherine Myers and I had developed and proposed a computational model of the hippocampus in which we argued that stimulus representations change during learning to alter generalization. And the basic idea behind this theory is that we suggested that the hippocampal region, is creating more similar representations in some cases so as to enhance generalization, and more dissimilar representations in other circumstances so as to minimize generalization. And these new representations, these altered or adapted representations, are then used by the striatum and the cortex and the cerebellum for various types of long-term memory. There's been a long tradition of thinking about the hippocampus as the site of the cognitive map. In our case, we'd argued that the cognitive map looks something like this, at least for a New Yorker like myself, with gross distortions based on my idiosyncratic views of the world, where differences in New York may be highly salient, but I can never remember the difference between Indiana, Illinois, and Iowa. They're all sort of compressed together. They are, in a New Yorker's mind, functionally equivalent. And there's this idea of being functionally equivalent, which is the basis for the behavioral paradigms that we use to test this theory. So the paradigm is called acquired equivalence, and is one that comes from the animal literature. In the animal literature, acquired equivalence has been shown to depend on the hippocampal region. And it involves training an animal that two stimuli that look nothing alike initially acquire a functional equivalence so that they are treated the same by the animal by exposure. So let me show how we apply this animal paradigm to humans. So take the example, consider two people who look nothing alike. Okay? They're of different stature. They have different charming foreign accents, and they definitely go to a different barber. No reason to expect any similarity. And yet, over the last few months, as we've gone on this recruiting of new faculty, we keep going back to May's Restaurant and Robert Treat Hotel. And the first time we went, they had a special. It was either blue fish or green fish. And I noticed that Bart and Denis both chose the blue fish. And this was interesting, because we came back to May's again and again. And when we came back to May's the next night, then they had red fish and yellow fish as the options. And Denis ordered the red fish, but Bart had stepped out, and I had to order for him. And in a leap of faith, I formed an equivalence class on food preference between them, and I suggested that Bart would like the red fish, and indeed, when he came back, he was delighted by my choice. So how can we test that this really depends on the hippocampal region? What brains have hippocampal damage? Well, one source of hippocampal damage is in people who are at the very earliest stages of Alzheimer's disease. And in work with our colleagues at NYU at the Alzheimer's Center, we've looked at patients with mild hippocampal atrophy. These are non-demented elderly, so they're not diagnosed with Alzheimer's, but these people with it are at heightened risk. They're not definite, but at heightened risk for getting Alzheimer's disease. And you can see on the left that those with hippocampal atrophy can be seen from a structural image. Now, these people at this point have no obvious memory deficit. A standard declarative recall task shows that whether they have this atrophy or not, in these non-demented elderly, they're completely normal. So these are healthy individuals with a little bit of hippocampal atrophy. And we asked, could these sorts of acquired equivalence generalization tasks give us a window into some of the functioning of what's going on? So what we found is that those with hippocampal atrophy and those with no hippocampal atrophy, shown on the left, were totally made the same number of errors in terms of the learning phase. But on the generalization phase, where they had to essentially guess what Bart wanted for dinner, those with hippocampal atrophy made way more errors. They failed to make the appropriate inference. Now, you might say, well, maybe these people simply forgot what they had learned, but we interspersed retention trials, and they do fine. So the idea is these people with very mild hippocampal atrophy, not enough to cause memory deficits, are fine at learning, they're fine at retention, but they're impaired at using their learning to make a leap of faith, to show an acquired equivalence for the stimuli that have had a functional similarity in terms of their outcomes. So once again, we asked, now how can we identify prodromal cognitive deficits in people who are at risk for Alzheimer's? Another way of going even further back. And for this, we ask, where do we find prodromal patients, people who are at heightened risk for Alzheimer's? And this takes us down to South America, to Antigua, Colombia, where there's a small rural village where the people come primarily from the Basque region of Spain. And one of their ancestors had a particular genetic defect that causes a very high penetrance of Alzheimer's, the familial form of Alzheimer's disease that would often develop in their 40s or 50s. So with these people, in collaboration with John Ringman and with my postdoc, Jesse Simon, we compare young, healthy, asymptomatic mutation carriers, so perfectly healthy young people, and their non-mutation carrying kin who are also healthy. And what we find is that the gene carriers are normal at learning, but impaired at generalization, just like those with hippocampal atrophy, while the non-mutation kin are normal at both. So even without a gene test, we can do pretty well at determining who in these families carry the gene or don't. So let me summarize. The hippocampal region modulates generalization by changing representations during learning to increase or decrease similarity. That was our computational model. Non-demented elderly with hippocampal atrophy are normal at learning. And generalization deficits are apparent even in healthy gene carriers of rare familial Alzheimer's. So some of the current directions in Alzheimer's that we're sort of building on this include looking at gene behavior links in the familial Alzheimer's disease patients, looking at cognitive assessments in rodent models of these tasks. These tasks came from the animal world. We can go back and use them in the context of drug discovery for Alzheimer's. and looking at some of the cognitive correlates of some of the new brain imaging biomarkers for Alzheimer's disease to relate these brain imaging biomarkers in healthy individuals to some of these kinds of memory deficits. So that brings to the end of the first part of the talk, these two main threads. And now I want to show how these two threads, the hippocampal generalization and the striatal learning, come together in three new directions that I want to tell you just a little bit about each. So the first is mood and anxiety disorders, studies of major depressive disorder and post-traumatic stress disorder. So the questions, since striatal and hippocampal dysfunction has been implicated in major depressive disorder, also known as clinical depression, we can ask, how is feedback learning and generalization affected in major depressive disorder? And how do antidepressant medications alter these cognitive functions? So analogous to the kinds of questions we asked about Parkinson's disease. So where in the world do we find depressed patients who have not yet been medicated? Certainly not in this region, where just about anybody who's not smiling is put on Prozac by their doctor. So very, very hard to find, any psychiatrist you talk to, very rare to find them seeing anyone who comes to them who hasn't first been put on Prozac by their general practitioner or nurse. Well, one place we can find it is in the West Bank and the Palestinian territories. There's about 2 to 3 times the rate of depression as the US. About 36% of the population suffers from depression, which means there are over 1 million Palestinians in this region who are diagnosable as clinically depressed. But in this region, there's a great degree of underdiagnosis and undertreatment, and a large numbers of these patients have never been treated. So this gives us an opportunity both to have a lot of patients who are depressed and particularly who've never been medicated before with antidepressants. Our work in the West Bank is in collaboration with Al-Quds University Medical School, which is in a suburb of Jerusalem, just inside the West Bank. And this project began in 2005 when I was visiting the university, and I met with Adel Misk, seen there on the left, who's one of the leading neurologists in the West Bank. And he told me of his dream to want to sort of build up neuroscience in the West Bank, where there was essentially no neuroscience research at all. And I told him if he could find 3 of his students, 3 students who would work with us, we could try to get a project going, a pilot study. And initially, these are all the students he found, none. None of the students, the medical students are very busy, and the students are very skeptical, and they really thought it was sort of a crazy and silly idea and a waste of their time. But finally, 3 years later, he was able to find 3 students who were naive and gullible enough to actually devote time for no pay and no obvious outcome. And these are those 3 students. You can see them. One of the guys, the second from the left, you may recognize. There's a young 23-year-old medical student named Mohammed Herzela. And he soon took control of the whole project locally and organized everybody and pretty much became the engine of the entire process there. And by 2010, we had a much larger lab. By 2013, the lab has continued to grow. And where in 2008, 2005, we couldn't even find 3 students who would be willing to do it. But now there are 3 students who are in line for every position in this lab. And that's really due to Mohammed's leadership and organization and his rigor in recruiting and selecting people. Working on this project has had many benefits for me, scientific and otherwise, but really the biggest benefit has been getting to know some of these students, and I'd like you to meet some of them. Oh, wait, we don't have the, where's the sound? Okay. So I'd like you to meet some of these students, see if this works.
Like 10 students, we were selected from 27 students, candidates. So I was the lucky one because I ranked number 10.
Ever since I was little, I wanted to discover something new, and my name will be remembered after discovering it. That's like you could say, a holy grail or something like that. Something I want.
I entered the medical school because I believe the most noble thing you can do is to relieve patients' suffering. But when you understand how really the disease happens and the aspects that it affects, you're not treating an individual, but this way you can help the whole society.
So with Al-Quds, we have a memorandum of understanding between our institution and theirs for a collaboration that sends faculty and students back and forth in both directions. And with a grant from the NIMH, from their brain disorders in the developing world, we have the resources to begin to answer those questions that I started off with. And the way it works is these students go out to clinics all around the West Bank, many of them sort of small local clinics in the cities of Jenin and Nablus and Ramallah, and test the patients, work in the clinics, test the patients. And so far there are 2 papers on major depressive disorder, one on learning from negative feedbacks in patients, and the other on how depression affects learning and SSRIs affect generalization. A brief summary of sort of the results is that in learning we find that depression impairs the striatal components of these tasks, but that SSRIs improve it. They remediate that deficit. In contrast, in the generalization tasks, we find that there's no effect of generalization in those who are de novo depressed, but once they're put on SSRIs, it creates a new impairment. So just like the dopamine agonists create new impairments in cognition in Parkinson's, SSRIs seem to create specific generalization impairments. For those of you who'd like to know more about this study, I invite you to come back here, same room, at 10 o'clock on Monday, when Mohammed will be proposing his thesis and describing this work in more detail. So some of the current directions are looking at some of the cognitive and genetic markers to identify at the de novo state who will and will not respond to SSRIs, very clinically relevant because only about half people do, and to understand the time course of these changes. Some of our future directions are looking at the cognitive side effects of SSRIs and how they compare to other treatments, looking at the effects of SSRIs in healthy non-depressed individuals, and looking at some functional brain imaging correlates of the cognitive changes due to SSRIs, some projects that are in collaboration with Catherine Harmer at Oxford. Let me talk now about the other aspect of mood and anxiety disorders, post-traumatic stress disorder. And a traditional view of PTSD is to think of it primarily as an anxiety disorder, one of which is focused on the frontal lobes and the amygdala. But growing evidence suggests that one can also view PTSD, post-traumatic stress disorder, as a learning disorder, and consistent with the idea that hippocampal involvement has been implicated in PTSD. And what role might the hippocampus be playing in PTSD? And we've suggested from our computational models of the hippocampus and from rodent work that one can think of the hippocampal role in PTSD the following way. We know we don't need the hippocampus for tone shock learning. The rat can learn it even without the hippocampus. But you do need a hippocampus for context-mediated learning. A rat needs a hippocampus if it's going to learn to distinguish that the tone in the black box means it's being shocked, but the tone in the green box means it's safe. And this, we suggest, may be a metaphor for what happens in the hippocampal involvement in PTSD. A soldier learns last year in Afghanistan that a loud noise is a life-threatening predictor, but then overgeneralizes this year in Washington, D.C. or in Newark, and so they basically make the right response in the wrong context, the wrong time and place. So in other words, they fail to have a contextually mediated discrimination of what a fear-predicting cue is. Where in the world do we find lots of people with PTSD? Well, not so far away from the previous study in Israel, where we've been collaborating in past years with the Jerusalem police, and in particular with some of the units that are at most high risk, the Victims ID unit, the ones who go in and identify body parts, and the bomb squad, the folks shown on the right with their bomb robot, and working with them and testing them. And also back to Hungary, another now a 2-hour road trip out of Budapest to another rural region that had a horrible toxic flood of a waste dump that destroyed several villages. And comparing these two populations, we were able to show that on the same task that I showed you before, this generalization task, that individuals with PTSD show a selective deficit in generalization. And we see that both in a civilian population in Hungary and in a paramilitary population in Israel. Also in the Middle East, looking at another population at risk for PTSD are Marines who've been exposed to blast from bombs, and in collaboration with the Navy, working at Camp Leatherneck to screen people who've just been exposed to blast in their operating bases to identify who might or might not be most at risk for both PTSD and depression. So our current PTSD research has 3 questions. We want to characterize the learning and generalization deficits in diverse PTSD patients. It's a very heterogeneous disorder. We want to assess military and paramilitary personnel prior to deployment for the prospective risk factors for PTSD. And in collaboration with Charlie Marmer at the NYU Medical Center, looking at biomarker correlates of some of these cognitive changes in those with PTSD. One of our future directions that we're beginning to start now is to look at the use of these cognitive tasks to assess and predict treatment efficacy. And our hypothesis is that generalization scores pre-therapy will correlate with the efficacy of cognitive behavioral therapy, which is one of the most common and treatments for PTSD, but one that only works in a fraction of the people. Now, why should this be the case? Why should these cognitive assessments predict outcome? Because we can think about therapy as generalization. So consider someone who's highly dysfunctional in their daily life. Homer Simpson, you're all familiar. Imagine Homer Simpson goes to the therapist for cognitive behavioral therapy. He gets all the skill training for the 12 weeks that he would normally get. But how he does in that therapy office isn't really the measure of success. It's what happens when he goes home to Marge and the rest of the family. Can he generalize the skills that he learned in cognitive behavioral therapy to the rest of his life? And so one of the studies we want to get going is to see whether we can predict which people will or won't show benefits based on these kinds of cognitive screening. So let me briefly tell you just a little bit about 2 other projects. We have behavioral neurogenetics with a new grant with Jesse Simon on the effects of genetics on reward learning generalizations of associations in aging. We're particularly looking at two genes, the DAT1, which affects the caudate and the striatum, and the BDNF and the hippocampus on generalization, and conjecturing that aging will magnify the effects, that these genes affect various aspects of learning and generalization, and we expect as cognitive resources decline with aging, that the effects of these genetic differences will be magnified. In the Palestinian territories, we're also looking at genetics, particularly looking at dopamine and serotonin genes, both looking at variability in healthy individuals, as well as the effects of as well as a predictor of the cognitive effects of medications in both Parkinson's and depression. One of the reasons why the West Bank is particularly a useful place to do genetics research is there's a lot of racial and genetic homogeneity as compared to the diversity of Rutgers and the environment here. And there are a number of families who have consanguineous families with intermarriage that have brain disorders. And in particular, a lot of our work has focused, at least in the healthy normals, in the southern part of the West Bank around Hebron, which has traditionally, dating back to the Ottoman era, had many fewer transportation connections with the other parts of the Middle East, and the people are much more inbred than they are in the rest of the West Bank. So even within the Palestinian community, we're going to a narrower subset where we find even more homogeneity. So let me lastly turn to talk about lifestyle influences on brain health. How is it that any of this that we've been studying is of useful to us in our daily life? And we have two threads there, looking at both sleep and looking at brain health in the African American community. With a grant from the NSF Smart and Connected Health Program, we've been looking at long-term mobile monitoring and analysis of the sleep cognition. In particular, looking at multi-day studies where people are in their own home, and looking at the effects of REM and non-REM stages on different aspects of both learning and generalization. And what makes this project unique is that it's using some of the newer technologies, the mobile wireless at-home methods. So traditional sleep research is done in a hospital or in a medical lab with a standard polysomnography where people have to come in, and it's a very unnatural setting to try to get a night's sleep. Instead, we've been using a lot of these mobile technologies that allow us to go into people's homes, set them up to monitor their sleep and monitor their cognition, and allow us to do long-term, day, week, or even several week-long studies of the sleep-wake cycle and how it affects cognition. So let me end up by talking about some of our work in the African American community. This is the final stop on the global tour, and for those of you who've gone on long trips, you always know the last stop is always the sweetest. It's the one that brings you back home, in this case back home to Newark, New Jersey. And let me just give you a brief overview of this project. It's the African American Brain Health Initiative. It's a partnership both within the campus between us and the Office of University and Community Relations, and between the university as a whole and the outside community. And it's motivated by the fact that African Americans have twice the risk for Alzheimer's disease and memory loss due to a variety of lifestyle, diet, and behavioral problems. Our project leverages some of the strengths here at the Neuroscience Center to ask how can we take some of what we do and contribute to the local community in which we are embedded. And we integrate really sort of three of our missions of the university: the community engagement, research, and education, all coming together and working in partnership. I've shown on the right there some of the partners, a dozen or so of the partners, churches, senior centers, city and state organizations that we work with here. And we've been doing this for about 8 years, and up until now it's always been a community engagement. It's been a community education, an outreach program. But these trust that we've built up over the last 8 years gives us a basis for connections in the community to begin to develop new research programs that look at pathways to healthy aging in the African American community, and also to allow us to take students from the Newark schools who come to Rutgers, train them and send them back through a program we call the Newark Brain Health Scholars Program that allows Newark-raised students to go back into their community and be trained in biomedical research. So over the 8 years, we've had a variety of programs: brain and health programs for seniors, lunch and learns, dementia caregiver, brain and heart healthy soul food cooking, which is the photo in the middle. And all of this has laid the groundwork for our now to begin to come back to sort of the research agenda for the CMBN on a program that has two arms. The first arm is observational studies. I described before how we have these genetic projects that look at how these various genes affect learning and generalization across the lifespan. There's obviously genetics doesn't determine everything. It's not entirely our destiny. And so we want to ask, particularly in the African American community, how do factors like obesity and cardiovascular fitness? And we have a collaboration with Brandon Alderman, a professor who's a professor from the Department of Exercise Science in New Brunswick, who works with us on integrating the physiological exercise physiology with cognition to understand these factors. And interventional studies, it's not enough to figure out why some people do better or worse with aging, but what can we do to improve those who are on a worse trajectory? And with Brandon, we're setting up some exercise and yoga classes to see whether we can change those who are most at risk for cognitive decline, whether it's frontal, striatal, or hippocampal decline, see which of those we can change with exercise or yoga. One of the big barriers is the fact there's those people who've done any NIH grants or FDA know there's always a mandate for diversity in recruitment, and it's traditionally, whether it's for NIH grants or FDA drug trials, it's always a challenge to try to recruit African Americans to studies, because historically there's a lot of suspicion because of historical abuses like the Tuskegee syphilis study. So one of the things we have to do before we can do the research is go out into the community, explain the research, explain the safety measures, and explain why it's important for people to participate. And this is sort of the main study we have, and we have this flyer which we printed up. And we have with our community advisory board, and people like Deacon Dixon up here in the third row are critical for sort of advising us on sensitivity to the community's needs and making sure that everything we do is sort of culturally appropriate. And this flyer, which we had printed up several thousand, we distributed them. On Tuesday, we had one of our big community events. And I got a message that one of the ladies sitting in the table wanted to see me. She was very upset because our flyer was discriminatory, and she was livid. And I'm thinking, how could it possibly be? We had our community board look at it. I know that Deacon Dixon would never let anything go past his eyes that anyone would view as being inappropriate or discriminatory. So I went over there, and I said, yes. And she says, this flyer, she says, it's discriminatory. She says, I'm 91 years old, and I'm sharp as a tack, and I could beat anyone at this table on a memory task. So we no longer discriminate against 91-year-olds, and we include them in our study. So if anyone wants more information, here's a website for this particular program. So I've taken you on a tour through some of our threads of research on the striatum, on the hippocampal region, some of the new integrative directions. And I'd like to just sort of briefly go back to Aristotle's question and ask, what have we learned in the last 2,000 years? And sort of review what I think are sort of the main take-home messages from the talk that I've given here. So an interdisciplinary overview that will span the philosophy, psychology, neuroscience, and clinical directions. We started with the philosophy of contiguity and frequency as Aristotle's principles of association, which led to theories of learning and stimulus-response learning from reward and punishment, to focus on the striatum and the basal ganglia and dopamine, with clinical implications for Parkinson's and depression. These then provide insights to the medical treatment and some of the effects of some of the interventions, medical interventions, the drugs, which allow us then to understand how these drugs might affect the neuroscience that are disrupted here and impact learning. The second pathway starts with Aristotle's principle of similarity, which we have mapped onto generalization and the generalization from adaptive representations, which associate with the neuroscience of the hippocampus and the medial temporal lobe, with implications for Alzheimer's, depression, and PTSD. And again, this not only provides us sort of a line of basic research, but sets us up to ask questions from a medical and clinical perspective. When we treat people for these disorders, are we affecting the hippocampus? And if so, how might that be altering generalization? So thank you very much. That brings me to the end.